Hello, you are welcome in biology classes organized by Society of Career Technology. Today we will talk about one topic of class 9th, the classification. Before coming to the point, I would like to say you that in 1967, when I had started my career, I had read a book on animal diversity. It means that time the concept of animal diversity had come. But it is surprising that after 50 years, we are talking about 5 kingdom classification. And it becomes very difficult, very cumbersome for students in the beginning. I suggest you that you have to start from animal diversity and then it will become very interesting. Of course, in biology, you have to learn many new technical terms and then about its characters. But let it be reverse. First of all, we should consider the characteristics and then what the terms are used for this. So, in diversity, millions and billions of animals and plants are found in this universe. There are plants, there are animals. In the animals, we have some flying animals, there are some crawling animals, some aquatic animals, some desert animals. And for living in different conditions, they develop different characteristics, and for that different characteristics, we use different terms. So if we start from animal diversity, then this topic becomes interesting. The earlier time we were taught about 2 kingdom classification, the plant kingdom and animal kingdom. But later on we came to know about some organisms which were not exactly identical to either plant or animal. For example, fungi, some bacteria and some unicellular organisms. So in 1965, Whitaker, a scientist, used Whitaker 1965. Although you will be surprised to know that in 1990, another concept has come, 6 kingdom classification. In competitive examination it is asked, but I don't know why in the syllabus still we are talking about 1965 Whitaker's 5 kingdom classification, and the concept of 6 kingdom classification has not yet been included in syllabi of many systems, ICSE, CBSE and UP board. This is funny, this is ridiculous. Anyway, as your syllabus contains 5 kingdom classification, I start from this very. Actually, all living organisms have some molecules. Water molecules are there, amino acids are there, and then nucleotides are there. Nucleotides are formed by some nitrogenous bases, phosphoric acid H3PO4 and 5-carbon sugar. The three combine to form nucleotide. So the first organism, which is actually connecting link in between non-living and living, had nucleic acid, DNA or RNA, which was a polymer of nucleotide. So the simplest organisms are viroid. Viroid have either DNA or RNA, but no covering, but it transmits certain diseases to certain organisms, and it can divide. If it enters in a living cell, it can multiply. Then some viruses developed which had a covering or capsid along with this nucleic acid core. They have had the capacity to itself, they were not living. They were connecting linkage between non-living and living. But when they enter in a living cell, they start multiplying. And they are very important. They are bearer of many diseases. You will be surprised to know that although we talked about that nucleotide and nucleic acid DNA and RNA are essential for life and for multiplication, but there are some prions. Prions have simple capsid but no nucleic acid, and it also transmits certain diseases. When it enters into some living organism, they also start multiplying. So this is a mystery how it happens. So in the 5 kingdom classification, these viruses are not included, but they are simply connecting link in between non-living and living. Now let us come to the 5 kingdom classification of Whitaker. He has divided a living organism into 5 kingdoms. The first of all, Monera. Monera includes the bacteria. They have a cell but no nuclear membrane, and simply DNA is there attached with this cell membrane. Some bacteria are beneficial for human being, some bacteria are harmful for human being. So all the bacteria are kept in kingdom Monera. Kingdom Monera includes prokaryote. Pro means primitive, karyo means nucleus. So here the nucleus is simple because there is no nuclear membrane, only DNA is there, no histone protein is there. Now, Carl von Woese gave in 1990 the concept of 6 kingdom classification, in which he separated some of the bacteria separately into a group, Archibacteria. Archibacteria, the cell wall was different and there was difference in the nucleotide sequence of 16S ribosomal RNA. Mind it. The two Archibacteria and Eubacteria are different from each other in the sense that the sequence of nucleotides in the 16S RNA is different in both of them. So they are kept in a different kingdom, Archibacteria. Then we have second kingdom is Protista, the unicellular organism. These are made up of a single cell with a nuclear membrane covering the nucleic acid. So they are called eukaryotes. There may be many algae and there may be many protozoals, but only thing is that these are unicellular or acellular organisms having a single cell. Then we come to know about organisms which are made up of many cells, which are called eumetazoa or metazoa. Here also we have a third category, the fungi. The fungi which are made up of many nuclei, and sometimes a word cenocytic is used for this. So they are separated from either plant kingdom or animal kingdom. They are kept in a separate kingdom, fungi. Now what happens? Then there are kingdom Plantae, in which in the cell we have a cellulose cell wall and there are some chloroplasts. So kingdom Plantae include all the multicellular plants. Then kingdom Animalia, containing many cells, and they include all animals. Now, in this kingdom Animalia, we have very interesting things. Let us come to talk about those interesting things earlier and then about their detailed numbering, naming. Initially, the primitive animals were like a glass or a ring, in which any cut passing through the center was dividing it into two equal halves. So these type of organization is called radially symmetrical. Initially the animals were radially symmetrical. They were either like a glass or like a ring in which any plane passing through the center was cutting it into two equal halves. Actually the cynentrates, the first group of the kingdom Animalia, are radially symmetrical and they are diploblastic. Diploblastic body made up of two layers, outer ectoderm and their inner endoderm. You will ask, what about sponges? Sponges are kept in another group, phylum Porifera or Parajua. They are not in the direct line. They have many cells, but there is no tissue formation. You know, tissues are group of cells performing one common function. So tissue formation starts from here, cynentrates. Here we have many cells, but there is no clear-cut tissue formation. The sponges are interesting in the sense that they have many cells, but there is no tissue formation. They have many pores on the surface called ostia, through which water comes in, and there is one opening at anterior end called osculum, with which water goes out. And most of the sponges live in oceans. They are marine, but only 3 species are there which are found in fresh water. They are Spongilla, Aphidacia and Minia. Now about these multicellular animals. Initially the animals were radially symmetrical. After that they became bilaterally symmetrical, as we are externally. Although internally we have alimentary canal single in one side, heart and one side, so perfectly we are not bilaterally symmetrical, but externally we are bilaterally symmetrical. Only one section passing through the middle cuts us in left and right equal halves, and these animals are triploblastic, having 3 layers in the body: ectoderm, mesoderm and endoderm. Now let us talk about these triploblastic animals. Triploblastic animals are of 3 types. Initially the acoelomate. In acoelomate animals, the mesodermal cells was filling the space in between outer ectoderm and inner endoderm. Endoderm was making the alimentary canal. Example of acoelomate is phylum Platyhelminthes, the flatworm or tapeworm. Then we have pseudocoelomate. Pseudocoelomate, we have mesodermal cells in the middle, but they are scattered. There are much more intercellular spaces. So there is a false cavity in the middle, which is called pseudocele. So these animals are called pseudocoelomate. For example, Nematoda, Ascaris. Now one more term you have to listen. In some of the animals, this mesodermal cells were arranged in 2 lines, some around this endodermal cells and some just below the body wall. So a cavity developed in between. This cavity was surrounded from mesodermal cells from both sides, both from outer side and from the inner side. So this cavity is called true coelom. So coelom is a cavity which is surrounded by mesodermal cells from both sides, and these animals are called coelomate. Then these coelomate animals were again divided into 2, protostome and deuterostome. At gastrula stage, when there is a blastopore, in some of the animals this blastopore makes mouth and anus is formed at the just opposite end. These are called protostome or schizocoelomate. In some other animals, this blastopore end forms anus, and mouth is formed at just opposite end. Here in protostome, blastopore opening of gastrulate stage makes mouth, anus is formed at the opposite end. But in deuterostome, mouth is formed at the just opposite end and blastopore area forms anus. So these are called deuterostome. The protostome or schizocilomate include phylum Annelida, Arthropoda, Mollusca. And then deuterostome or enterocilomate includes phylum Echinodermata and Chordata. Although when you will reach in higher classes, you will come to know about separately Chordata and Vertebrata. Chordata and Vertebrata are not the same. But in your book of 9th class, it has been written Chordata or Vertebrata. Chordata having a notochord for some time or for full time in the life cycle, and Vertebrata which have a vertebral column in our body. So the animals which have a vertebral column, it's called vertebrata. So initially the animals of invertebrates were not having vertebral column. They were very small. They were like earthworm and cockroach touching the ground. So that any change taking place in the ground was much more affecting them. So the nervous system was on the ventral side. But when the animals became much more large, what happened? They developed some hand appendages, hand, legs, etc. Now, which part of the body will be affected much more from environmental changes? Obviously, this area. So here we developed a dorsal nervous system, and later on it was changed into vertebral column, and the lower animals had a ventral nervous system. So this is one very important thing. Initially the smaller animals had ventral nervous system, but the higher animals developed dorsal nervous system. Then these dorsal nervous system having vertebrates were again divided into 5 groups. One, the Pisces or fishes, which were aquatic animals. Then amphibia, frog, etc., which could live inside water also and outside water on land also. The reptilia, for example lizard, became modified for total terrestrial habit, dry land. Of course, some of the reptiles, crocodiles, etc., again entered into water, but their skin is modified for living on dry land. Then the aves or birds, which develop the capacity of flight. Then mammals, in which we are included. Again, I would like to tell you a very interesting thing. In birds, what happens? Birds need to have very less weight during flight. So for reducing the weight during flight, they have developed some very interesting fact. They have single ovary, no urinary bladder, no penis, no clitoris. For example, pigeon, hen, etc. No teeth. Some of the birds which are flightless, for example ostrich, of course they have urinary bladder, they have penis, they have clitoris, and of course they don't have teeth. Only Archaeopteryx, which were the connecting link between reptiles and birds, the fossils, they were having teeth. One more interesting bird is there, Fishes, amphibians, reptiles, they are cold-blooded animals. Their body temperature changes with changing environmental temperature. Birds and mammals, they are warm-blooded or homeotherms. Here the body temperature is maintained even environmental temperature changes. Then one more word, fishes and amphibians are anamniotes. They don't have amniotic cavity, but reptiles, birds and mammals have amniotic cavity around developing embryo. They are called amniotes. So I would just wish to emphasize you that you have to learn the different characteristics in animal diversity in the beginning. Later on you have to learn one by one some new technical terms. If just in the beginning you will be taught about all technical terms in the beginning, it will become cumbersome for you. So I have made an experiment. I wish that it will become interesting to you. Thank you very much.